So in this lecture, we're going to talk pretty extensively about wound healing. Wound healing can be classified as tissue repair. We're going to see two different processes, and we'll compare and contrast them, much like we did acute and chronic inflammation. One process is going to be a healing response, and the second process is going to be a regeneration response. And I'll kick it off by saying our regeneration response in our body, as we get older, diminishes. Okay, it goes down over time. And at your age, it's probably difficult to see that. And about every decade, it appears that patients or individuals recognize that things are actually changing, just so you know. And so if you think about a decade ago, okay, let's say that you're 20, think back to when you were 10. Okay, when you were 10, if you fell down and cut yourself or hurt yourself, I mean, it almost seemed like it repaired in a day. It was kind of crazy. In fact, there might have been a wound when you were 10 years of age, a decade ago, that you swear that you flew off your handlebars, you endoed off, you hit the asphalt, you scraped across the asphalt, and it was gnarly, and you have trouble remembering which leg it was on because you can't really find it. And in contrast, maybe you had a paper cut like last weekend, and it still won't close. Okay? So our wound healing response as we get older changes and goes down significantly. Now you contrast that, go, instead of back 10 years, go forward maybe like six decades. Okay, let's say when you're 80 years of age. So if you interact with patients that are elderly, okay, or maybe grandma and grandpa, okay, or maybe friends of the family that are in their 80s, their wound healing response is very, very compromised. It's so compromised that they have to be careful in a hospital stay or in their wheelchair, or maybe even at home if they're bed rest, that if they don't get enough circulation, they're going to end up with bed sores. Okay? So you think that these seats are uncomfortable, but your young rear ends can handle it, right? But if you were in a soft bed for a couple of weeks as an 80-year-old patient, you likely generate some sort of ulceration between that interface, even though it's a soft bed. So just keep in mind that this healing process that we're going to talk about is designed in a certain way, but it doesn't necessarily follow all of these steps for our lifetime. Okay? And it gets worse as we get older. Okay, my clicker's working on this presentation, so maybe it didn't like the last time. So this is a graph showing a lot of the same things that we've already talked about in our inflammation lecture. But I wanted to show it one more time in a slightly different format. It's an older figure, but we use it all the time. And what this figure is showing is it's showing relative time on the x-axis, and then a relative percent of the maximum response. And it shows in A, which are these two first peaks, it's showing early inflammation followed by late inflammation. In early inflammation, what cell types do you think show up? Neutrophils. Do you think that's an important cell type to remember when it comes on the scene? Okay, because I've said it like, what, 5 times? What's that?
So it's not important, right?
It's not important, yeah. It's important that you remember it. Just memorize it, that's fine. The second peak, that's late inflammation, what cell types do you think show up there? Macrophages. What cell types do you think are present, though, in late inflammation? Any of them, in any composition. What do you think might be there? Acute or chronic inflammation? Oh, I thought I had the answer, now you confused me. So in late inflammation, what cell type do you think shows up? You just said it. The macrophage, the activated tissue macrophage. Do you think there might still be some neutrophils there? They just haven't left yet. Right. So you see how these overlap? That means as this one is fading and this one is coming up, you might still see some neutrophils in late inflammation. Now, if that second phase of inflammation starts to become early chronic phase, what are some cell types you might see? What are some of the cells that you would see in a chronic inflammation that might show up in a couple of weeks that macrophages stick around and elevate capacity? Foreign body giant cells, thank you. Okay? So I'm trying to weave what we just talked about in the last lecture into this lecture. Then in B, we see the events of B, which is granulation tissue formation. Granulation tissue formation is named because of the way it appears. So these inflammatory cells that show up start to release cytokines and growth factors. And those cytokines and growth factors recruit other cell types to the area. They recruit more macrophages, but they also recruit fibroblasts. They're also going to recruit blood vessels to come into the area. And so you get this region, this third peak of granulation tissue. It has a granular appearance to it. And there's still going to be, you can see how they overlap, elements of inflammatory mediators. But you're starting this remodeling phase by bringing in new blood vessels because that brings in nutrition to the healing tissue. And if you don't bring in new blood vessels, you'll never heal the wound. Okay? Then, at this point, you move into this phase known as collagen accumulation or tissue remodeling. Well, the fibroblasts are the cell type that manufacture the collagen. So you have to bring them in first before they can make the collagen. They come in during granulation tissue. They stick around after granulation tissue and they start cranking out new extracellular matrix, such as collagen. And this collagen accumulation remodels that tissue bed. And then post-collagen deposition or collagen accumulation, you have that tissue remodeling has taken place and you have resolution of the inflammatory response. It completely goes back to essentially baseline. And now the wound is considered to be quote-unquote healed. Make sense? So if you do that past the age of about 10, that remodeled tissue in this area looks very different than what it did before you were 10 years of age. 10 is kind of like that magic tipping point. And the reason for that is there's a whole host of genes that code for different types of extracellular matrix proteins. And the gene elastin turns off in your preteen days. So by the time you're 13, you've stopped really making elastin. And you're left with the elastin that you were born with. And all of your organs are pretty much organized with elastin content the way they're going to be for the rest of your life. It has a half-life of about 70 years. So when you're 70 or 80 years of age, you have about half the elastin you did when you were born. And elastin is a stretchy protein. It gives skin its ability to pull and snap back. It's also found in the vocal folds. It's also found in blood vessels, heart, bladder. Okay? Any organ or tissue that stretches is going to have an elastic component to it. It's going to have elastin in it. So as we get older, our voice changes slightly. Our elastin content in our vocal cords decreases. So they're not quite as able to stretch and recoil. And so that's why our voice slightly changes as we get into our geriatric years. It's also why our skin looks different when you're old. It's also why we have vascular disease and cardiac disease. Okay? It's also why you have to go to the bathroom multiple times a night when you're older versus when you're younger. So a lot of that stretchy of that tissue doesn't happen. So the magic thing that happens when you're less than 10 is that skin remodels and still has elastin that grows in there. And after age 10, approximately, and it's kind of a wax and a wane type of thing, you get a scar that has no elastin and it's just basically collagen. So it's stiffer, it doesn't stretch, it looks different than the original tissue. And that's really kind of the big reason for scarring in a wound environment. Okay? Okay, so what I want you to compare and contrast, and you can talk amongst yourselves, one of you pick A and the other one pick B. And I want you to talk about A, the difference between repair and resolution, and then person B, talk about the difference between healing by first versus second intention. Okay? So take some time to do that right now. Thoughts on resolution versus repair. Let's start there. Resolution versus repair. Okay. Did you guys hear that? So resolution is really the body's ability to restore back to function. It works the way it was originally designed to work. Okay, so if it's skin, you would have the return of not just organized skin, but you'd have the return of layers of dermis and epidermis, right? Because they have different functionalities. You would have the return of glandular structures like sebaceous glands. You'd have the return of hair follicles. Okay, so some of you, it's most obvious, I think, in wounds that happen on the head or the scalp, is if someone like had surgery when they were a teenager or in their 20s of the cranial cap, and you reflect back the hair, you can kind of see where it didn't grow back. So you can kind of see where that incision line was. Or people on their arm, maybe there's a region of scarring that doesn't have hair, arm hair that's grown back, or on the leg. So that's the most obvious with respect to skin. But if we looked at the heart, and you hear this all the time, so-and-so had a myocardial infarction, a heart attack. We think they're going to do well, but we just don't know how much scarring there was. That's what the doctor said. What are they talking about? Well, if it scars, then there's a fibrotic region that no longer has contractile properties because it's not cardiac myocytes. Or in a stroke patient, that fibrosis, which is a gliosis, is what we call it in a stroke patient, same thing. It's not new neurons that grow back. It's extracellular matrix that fills the hole, kind of like filling the hole on a wall. You move out of your apartment and you hung all these pictures, right? And let's say you did it really messy like and put big holes in the wall, and then you tried to patch it with that patch stuff from Ace Hardware or Home Depot, and your landlord's like, yeah, it's pretty obvious, I'm going to have to do some work there, because you can see where the patchwork is. Okay, so that's the scarring that takes place in the brain or the heart or in the kidney. Versus regeneration, right, or repair. Repair is that fibrosis resolution is where you have full healing or return of function. Okay, so that was quite good. How about first versus second intention? First versus second intention. Did you guys catch this in the reading at all? First versus second intention. Yes?
First is more like a surgical occasion.
Okay.
Where there's more approximation.
Yep.
And if it's a deeper cut or something that's deeper cut, but more damage and heavier.
Okay. So she said, for those of you who can't hear in the back, first intention is more like a surgical incision, where you can actually nicely approximate the margins of the wound. So if you cut it with a razor blade, right, or a scalpel, and you move it away, and then you bring it back together, and you approximate those tissues very nicely, that would be healing by first intention. So you're going to have a really nice result. Healing by second intention is where it's usually trauma, like the skateboard injury or the bicycle accident. So you've heard of plastic surgeons that go in and can revive scars. What are they doing there? Well, they try to go from a second intention healing to a first intention healing. And the way that they do that is they cut out the scar and create nice, clean scalpel edges and then bring it together. Okay, so if you had a wound back here, and this was all nasty, okay, they would come in, create sort of matching first intention scalpel incisions, and then they approximate these two together. Does that make sense? And try to essentially eliminate that second intention result. And so if you have a nasty scar that a plastic surgeon has looked at or said, you know, if that really bothers you over time, we can do our best to try to make it look better. It may never go away completely, but we can probably try to convert it from a second intention healing to a first intention healing. And the younger that you do that, the better the result. Okay? All right, so let's look at this figure, which further describes the two different processes of regeneration versus healing, okay, in a picture format. So if you've got a cell, you have an injury, you can go down a regeneration or a healing pathway. There's two other terms that we usually use to talk about this that mean the same thing. So healing is also repair, or regeneration is also resolution. So depending on what textbook you read or which literature you're reading in the peer-reviewed literature, someone might describe it as one word versus another. They really mean the same thing. So let's talk about the best way for it to take place, which is resolution or regeneration. And on this side, you're going to write down little notes like restoration of function. Okay? So for example, if you get an injury, you can completely regenerate and restore functionality. Or if you have a stable tissue and you lose a portion of it, it regenerates almost to its full extent and returns to normal function with very little to no scarring. So tissues that can do more of the regeneration in you and I are things like epithelium, our skin. Every 6 to 8 weeks, our skin turns over and you have a whole new skin. Okay? That's why you have to cut your nails, because that's part of that stratum corneum that's actually kind of sloughing out, right? So your fingernails and your toenail growth is kind of indicative of how quickly your skin turns over, or your hair as well. Some of us, it grows faster than in others, but these are complete regeneration. That 8 weeks later, it's not like your skin looks that much different than it did the 8 weeks prior. And the same thing happens in the GI tract. Same thing happens in epithelium throughout your body. Now, when you get older, that process becomes compromised, right? Because age is actually considered a pathological condition. So that normal process of regenerating skin in an 80-year-old patient is not what it is in this classroom. Okay? The liver and the kidney also have a great ability to regenerate. You've probably heard that. Oh, the liver has a tremendous regenerative capacity. And that's absolutely true. But there's a limit to which it will regenerate. And we'll talk about that in our hepatic section, our hepatic and renal section. But the liver hepatocytes can regenerate to a great extent as long as there isn't a lot of scarring. And we'll talk more about that in that lecture series. Now, if we look over at the less favorable side of the tree on the right, we see that not everything is going to regenerate. Not everything is going to resolve. Not everything is going to return to normal function. In the case of normal function for the liver, now you can filter again. You can go through lipolysis or lipogenesis. You can convert glucose to glycogen or glycogen to glucose. You can do all the normal functions of the liver, helping to recycle red blood cells. All of that returns. Filtering the blood. I think I said that. Same thing with the kidney, is you can concentrate urine. Same thing with the skin, is you have hair that returns back to the skin. Or sweat glands, sebaceous glands. Now over here, this doesn't always take place, and if the injury is large enough or the patient's older, you can't just leave this massive gap. And so what you do is you use a fibrotic response, you bring in tissue macrophages, they recruit fibroblasts, they spit out collagen, they lay down a new matrix. But that new matrix is like puttying the wall where you hung your picture when you moved out. And you can tell exactly where it was. Now, if the wound healing takes place post-chronic inflammation or during chronic inflammation, we get a tremendous amount of fibrotic scar woven into the tissue bed. And the case here that I want to point to in our hepatic section is going to be liver cirrhosis or cirrhotic liver disease. Okay, and you've heard that probably, that the liver regenerates unless there's scarring, and that's a cirrhotic liver situation. Okay, oops. All right, let's do this. Let's ask this possible test question. What's the role of fibroblasts during the reconstructive phase of wound healing? A, to generate, new capillaries with vascular endothelial cells around the wound. B, to establish connection between neighboring cells and their fibers, help to contract their fibers. C, synthesize and secrete collagen and the connective tissue proteins. D, provide enzymes that debride the wound of dead cells, or two of the above. Do you guys know what the word debride means? Or debride? It's a clinical term that means to clean out the wound of dead tissue. So we do it using enzymes or fluids. So we'll wash it out. We'll clean it out with maybe hydrogen peroxide, or we'll flush it with clean, sterile saline. Other cultures might use maggots, right? Because maggot therapy will come in and eat up the dead tissue and not touch the live tissue, so that's debriding or debriding the wound. Okay, so what's the answer to this question? C, synthesize and secrete collagen and the connective tissue proteins. Anybody else like another one? Two of these. So what are the two? B and C, establish connection between neighboring cells and help to contract their fibers. Yeah, I like that one too. So it would be B and C. Remember, that's that collagen accumulation, and that's the wound contraction phase that we went over in that diagram. Very good. So when we have injury, how do we know, or how does the body discriminate between when it forms a scar and when it doesn't form a scar? So one of the ways that it does this is, you can see on the left panel is regeneration, and on the right is scarring, okay, or healing. So if you've got, this is an hepatic lobule, so we've zoomed in on this box. You can see our portal triad right here, and we've subjected it to injury. And when we result in injury that's just to the cells, and it doesn't disrupt the matrix that the cells live in, these liver cells, these hepatocytes, are able to proliferate, migrate in, and completely regenerate a functional liver. In contrast, on this right side, when we have damage to the cells as well as the extracellular matrix, you disrupt the architecture. When those cells move back in, they don't know exactly where to go because the architecture is different. And so you get more scarring and fibrosis that happens in this hepatocyte prep or in this liver prep if you have damage to the architecture, the tissue or the matrix, as well as the cells. So what would be an example of when you would have one versus the other, you might ask? Well, damage to the cells might be when you have a hypoxic condition to the organ, but it's transient. It's over a short period of time, where the cells die and the matrix is really untouched. Well, in contrast, if you had, say, trauma to the liver, like in a car accident, you might have complete laceration of a portion of the liver, and now the architecture, the framework has been disrupted. Or, if you have hypoxia or chronic inflammation to the liver over a long period of time, how would that influence the architecture? Can you answer that question? Did you follow what I just asked? If you had hypoxia or chronic inflammation to the liver, how would that potentially cause changes and damage to the architecture? Chronic inflammation is the key word.
Necrosis.
What's that?
Necrosis.
You get necrosis, okay? That's the first step. You have chronic inflammation, what cells come in? What cells come in first? Very, very nicely done. Neutrophils. You guys seem like you learned that. Secondly, what cells come in after neutrophils? Macrophages. And if it's chronic, what do you see? Giant cells. Okay. And now, how does that connect to remodeling the architecture? What do those cell types do? If they stick around, right? And they don't go away, they don't resolve. Do they recruit? What's that? Do they recruit fibroblasts? They're going to recruit fibroblasts in the area. Are they going to recruit them for a short period of time or a long period of time? Long period of time. Are they going to crank out more collagen that's necessary because they stick around for a long time? That's where that fibrotic response comes from. So if you have liver under a chronic inflammatory situation, like an alcoholic who won't stop drinking, you're going to get constant remodeling of the liver, and that's where the scarring comes from. You get the remodeling because the macrophages stick around, and they're constantly causing tissue damage. Okay? Okay, you guys remember a figure that looks like this from a couple of years ago? Yeah? If you don't, you just nod your head. I won't really know. It's more of a rhetorical question. So this is a figure that's highlighting the cell cycle. Why is that important? Why is that important in healing? Because these cells that we want to move into an area to heal it, whether it's regeneration or whether it's repair, we need those cells to migrate in, to replicate, divide, which is where this comes in, okay? And then either to leave the area or to slow down their proliferation. So in order to signal these cells to the area or to get them to divide, we use three main processes. We've got autocrine, paracrine, and endocrine signaling. And all three are involved in tissue healing. And I'll give you examples of each. So an autocrine signal, an auto meaning self or same, auto. So an autocrine signal is when the same cell releases a signal and has a receptor that binds that exact same signal, telling it to do something. So an example of that in a wound could be an activated macrophage that's been recruited to that area. It releases an inflammatory mediator known as interleukin 2, or interleukin 6, or interleukin 8. It makes all three of them. It has receptors for that interleukin family that bind and trigger the cell to manufacture more inflammatory mediators. Why would that be important? Because those inflammatory mediators, if there's a large enough chemical gradient, it's going to be detected by a faraway cell. This is peregrine signaling. So the faraway cell, meaning somewhere away from the cell itself, has those receptors and binds that interleukin 2, 6, or 8 and says, aha, I am supposed to be in an inflammatory, an activated inflammatory stage. And so it gets recruited to go help out that secretory cell. Where does endocrine come into play? Endocrine comes into play when that inflammatory mediator makes it to the bloodstream, because the gradient is large enough that it's detected by cells within the bloodstream, say circulating monocytes. And those circulating monocytes that are seeing that signal now in the bloodstream know that they're supposed to migrate out into the tissue. And that was a couple of lectures ago when we talked about how this whole process of recruitment works. Right? So we've got these circulating monocytes that marginate to the edge. We got a controlled clot that takes place to help slow down, cause a little bit of stasis. We activate selectins, so they get a little tacky and they start to bind and then break. And as they bind and break and bind and break, they start to roll. Finally, they roll, they get stuck by integrin expression. And when that integrin expression is in its high affinity state, that flattens out that monocyte and it squeezes, a process known as diapedesis, through the endothelium. And now it chemotaxis to where it came from, which is this secretory cell. So we use all three of these in this process of inflammation or recruiting inflammatory mediators. Okay, this is a busy slide, but if you can explain all the different parts of this slide, then I feel like you have a pretty good understanding of what's going on. So this might be a good study tool, something to go over in SI. See if you can go up to a whiteboard. A window in your apartment or your bathroom mirror makes a great whiteboard. Okay? And it wipes off, and then you clean off the toothpaste and the smudges anyway. So it's like a double bonus. You clean your apartment and you study at the same time. And I just saved you money. You didn't have to buy a whiteboard. Okay? So draw this out. If you look at the components here, here's the epithelium. Here are integrins binding to a basement membrane. Now we're outside of the cell in the extracellular matrix, and we see these fibroblasts. Here, underneath the skin, is a blood vessel in the dermis. You can see the red blood cells inside, stacked up like little pancakes almost. And you can appreciate if we zoom in on the interface or the layers of the blood vessel or the basement membrane. And that's this picture on the left. And we see there's an architecture of matrix proteins that make up those two structures. And you can see that they're made up of collagen, type 4 collagen, proteoglycans. Proteoglycans hydrate the tissue because they bind water. So they help to make the tissue moist, proteoglycans. And then laminins. Laminins are this sort of cross-looking architectural pattern that's staggered throughout extracellular matrix, giving it structure and integrity. If we look at the fibroblast, which is this cell, and what it's connected to, which is something on the extracellular matrix, the way I like to explain this interface, which is blown up right here, here's the fibroblast of the cell nucleus, and here are integrins that are attached to the extracellular matrix. These cells are like children on a playground on a jungle gym. So they have to attach and hold on to something. So as they look apparently in a lot of pictures, like they just sort of float in space. They're not really floating, they're actually attached to the extracellular matrix. And if they want to move and migrate through that extracellular matrix, they literally have to grab onto stuff like a kid would be crawling across the jungle gym. And those attachments between their surface adhesion molecules and the outside environment is what allows them to migrate through. And macrophages, I saw a picture of a macrophage a couple of lectures ago. It was a scanning electron micrograph of the macrophage kind of stretched out. It was the picture right before the hip replacement, where we're looking at the pox that the macrophages make. So if you look back at your lecture slides, you'll see that that macrophage is stretching out. That material that it was stretching out on was coated with collagen. I didn't mention that in the picture. But it has to have something to crawl on. It has to have a surface. It doesn't just levitate in space. It has to have something that it's holding on to. Okay. The other thing that's significant about this is this interaction. So this is the cell. Here's the nucleus. And we know that the nucleus is physically connected to transmembrane proteins known as integrins. And they're connected by these actin cytoskeleton. So decisions that are made in the nucleus are controlling, almost like controlling a puppet with strings, are controlling things that happen at the cell surface. So if the nucleus says, okay, let's migrate, let's divide and migrate, then it moves the integrins via this focal adhesion complex to bind up to the extracellular matrix. These are the bars in the jungle gym. And it grabs onto one and releases the other, and then moves over here and grabs onto another, kind of like a kid swinging on a jungle gym. So that's how these cells are migrating through during this wound healing response. Whether they're fibroblasts, right, or whether they're activated tissue macrophages that are coming in. Make sense? It's not magic. It actually all happens intracellularly, and those signals are manifested extracellularly. Okay. Angiogenesis. So the cell cycle, being able to divide and proliferate. The cell inside connecting to the outside, that's the intracellular signals connecting to the extracellular environment. Number 2. Number 3, this is under the broad category of mechanisms that these cells use during the wound healing process. Number 3 is angiogenesis. So I mentioned earlier that if you're going to heal a tissue, you actually have to have nutrition in that tissue bed. And if you don't have nutrition in the form of blood flow, it will never resolve. It will never heal. Okay? It'll never regenerate. So the angiogenesis process begins by sprouting of a blood vessel. So you've got a blood vessel here. It creates a little sprout, kind of like a tree, grows a new branch or a new leaf. And then from that sprout, it moves out into a new capillary network, and then it matures and perfuses a new tissue bed. And mediators that come from activated macrophages are angiogenic mediators. Things like vascular endothelial growth factor. Things like basic fibroblast growth factor. These are all angiogenic mediators. Remember I told you earlier, a couple of lectures ago, that VEGF, or vascular endothelial growth factor, used to be called vascular permeability factor, VPF. So these are things that the macrophage manufactures in order to stimulate angiogenesis, and this is what happens during granulation tissue formation. Remember how I said that you get new blood vessels moving into the wound in order to provide it with nutrition. How does that happen? The macrophage releases the angiogenic mediator, causes the blood vessels to sprout and perfuse a new tissue bed. So here's a picture in sequence. Initially, right, we've got a wound in the skin, and it goes deep. This wound in the skin forms a clot, a fibrin clot. You get platelets that are trapped. It stops the bleeding. You recruit from the bloodstream neutrophils first. They come in. Secondly, in 3 to 7 days later, this is within 24 hours, days later, you get the neutrophils kind of dropping down in population, and macrophages start taking residence. You can see that the vascular elements kind of grow, bringing in new perfusion and new nutrition. Then, weeks later, we get regression, angioregression, where the blood vessel goes back to its normal state. You lose a lot of the macrophages that came in because they're not necessary anymore. You're not going to maintain a chronic inflammation response. And you prepared that wound with remodeled collagen or a fibrous union. And this is the scar that takes place. Now, if we look at healing by, that was first intention. Healing by second intention follows the exact same process. It's just every step is exaggerated because usually the gap is much bigger. By first intention, you can see that the wound bed is a much narrower slice. And then in second intention, you can see that there's a much bigger gap that you're trying to heal. But it's all the same steps. You bring in neutrophils. You expand the vascular network to bring in perfusion or nutrition. But you do it more aggressively because the wound's bigger. So you see in this picture, there's a lot more vascular elements than what we saw in the previous picture. And when you contract the wound, you get a greater defect at the surface of the skin and a more robust fibrotic response because you've had to heal or bring in more collagen to fill that gap. So healing by second intention on this slide, and healing by first intention on this slide. All the same steps, it's just second intention is a much bigger initial hole or gap. And so you have a more robust response with second intention healing. Questions? Okay. This is a skin ulcer. It's kind of gross, but it's on a foot of a diabetic patient. We're going to be talking a little bit about this after the break. I'm just going to set it up a little bit right here. If we take a look at this ulcer of this foot, this patient's a diabetic patient, unmanaged. They get these ulcers that last and persist for weeks to months, and they don't resolve. They don't go away. And there's all sorts of complicating factors associated with them that we're going to talk about. But the idea is the blood flow is compromised into this distal region of the foot. And it might be a carpet tack, or they might have kicked a portion of furniture and tore a little bit of skin. Didn't even know it because they can't really feel very well, their feet. They have what's called parathesis, which is this pinprick feeling, like this. Like if you sit on your foot or you sit with your legs crossed in a certain configuration, after a while the blood flow isn't going down distally, and so you get that sort of, my foot went to sleep feeling. So these patients have that all the time. That would be really annoying. Okay, so they cannot feel, and they have that when they feel anything, they feel that pinprick feeling. So that's early sign of nerve damage. That parathesis that you feel, that's your body's way of letting you know that the nerve is actually being starved of oxygen because there's no blood flow going to it. So it gets your attention by giving you that feedback, that signal. So you move your foot to a different configuration. However, in a diabetic patient where there's significant vascular problems, and we'll talk about that later in the semester, what happens inside the blood vessel, that blood flow doesn't get to the distal extremities. Okay? Just last night, I woke up, you guys ever done this? I woke up, I was so asleep, but I was sleeping on my arm, and I woke up and I couldn't feel like my pinky and my ring finger, right? So I went to scratch my face and it was like, oh, right? I was like, oh my gosh. And I was like, 1, 2, 3, 4, oh, 5, they're all there. And then slowly it came back and it first started tingling, right? And then it came back in full effect. But it was just last night I woke up in the middle of the night and I'd fallen asleep like an awkward configuration. I think I cut off blood flow to my hand and I was counting fingers at 2 in the morning. And I was worried that something happened magically and I lost 2 fingers. But fortunately, they're still there. So I think, yes. So we're going to talk about this after the break. We're going to pick up right here, okay? And we're going to show histology of this type of lesion in about 5 minutes or 10 minutes when we get back, okay?